Today, FDA approvals in bladder cancer and Hodgkin lymphoma, a breakthrough designation in AML, promising findings in kidney and breast cancer, a European approval in multiple myeloma, and a missed endpoint in a gastric cancer study. Welcome to Enclave News Network. I'm Gina Columbus. The PD-L1 inhibitor atezolizumab was granted an accelerated approval by the FDA as a treatment for patients with locally advanced or metastatic urothelial carcinoma whose disease progressed during or after platinum-based chemotherapy, or within 12 months of receiving platinum-containing chemotherapy either before or after surgery. The decision was based on findings from the Phase 2 IMvigor 210 study, in which atezolizumab demonstrated an overall response rate of 14.8% in patients regardless of PD-L1 expression. Among patients with PD-L1 expression of more than 5%, ORR was 26%. Complete response rates in the overall and high PD-L1 expression groups were 5.5% and 12% respectively. The Ventana PD-L1 assay was concurrently approved as a companion diagnostic. Dr. Sandra Horning, Chief Medical Officer and Head of Global Product Development at Genentech, the manufacturer of atezolizumab, said in a statement, "Tecentriq is a new medicine that can work with the immune system to treat people with a type of bladder cancer that progressed after platinum-based chemotherapy. We thank the scientists, doctors, patients, and their families who made it possible to bring Tecentriq to patients with advanced urothelial carcinoma." The accelerated approval is contingent on results from an ongoing confirmatory Phase 3 study, IMvigor 211, which is comparing atezolizumab with chemotherapy in patients with locally advanced or metastatic urothelial bladder cancer who have progressed on at least one prior platinum-containing regimen. The FDA also granted an accelerated approval to nivolumab for the treatment of patients with classical Hodgkin lymphoma that has relapsed or progressed after autologous hematopoietic stem cell transplantation and post-transplantation brentuximab vedotin. This marks the first PD-1 inhibitor that has been approved for a hematologic malignancy. The accelerated approval is contingent on the confirmatory outcomes of a Phase 3 study. The approval was based on a combined analysis from two single-arm trials, in which the objective response rate with nivolumab was 65% in 95 patients. Additionally, the complete remission rate with the agent was 7%, and the partial remission rate was 58%, while the median duration of response was 8.7 months. Adverse events led to discontinuation of treatment for 4.2% of patients, and 23% had a dose delay due to adverse events. Serious adverse events occurred in 21% of patients, with those occurring in more than 1% of patients, including infusion-related reaction, pneumonia, pleural effusion, pyrexia, rash, and pneumonitis. A breakthrough therapy designation was granted to CPX-351 by the FDA as a treatment for patients with therapy-related acute myeloid leukemia, or AML with myelodysplasia-related changes. CPX-351 is a liposomal formulation containing a fixed combination of cytarabine and daunorubicin. The designation was mainly based on Phase 3 findings, in which CPX-351 significantly reduced the risk of death by 31%, compared with cytarabine and daunorubicin for older patients with high-risk secondary AML. In the study, the median overall survival for patients who received CPX-351 was 9.56 months versus 5.95 months with daunorubicin. At 24 months, 31.1% of patients enrolled in the experimental arm remained alive, compared with 12.3% who were treated with daunorubicin. Complete response rates with CPX-351 and daunorubicin were 37.3% and 25.6% respectively. In regard to safety, no statistically significant difference was observed between the two arms. Full findings from the study will be presented next month at the 2016 ASCO annual meeting. The Phase 2 Cabo-Sun study in renal cell carcinoma showed that cabozantinib significantly improved progression-free survival versus sunitinib in treatment-naive patients with advanced disease. While full results will be submitted at an upcoming medical meeting, the drug's developer, Exelixis, did state that the safety profile for cabozantinib in the study was similar to previously reported outcomes with the treatment in advanced RCC. Exelixis is communicating with regulatory agencies about a potential first-line cabozantinib indication in RCC. Cabozantinib was recently approved by the FDA in April 2016 as a treatment for patients with advanced RCC who have received prior anti-angiogenic therapy. In a statement, Dr. Tony Choueiri, Clinical Director of the Lank Center for Genitourinary Oncology at Dana-Farber Cancer Institute and Chair of the CABOSUN study, said, "The positive outcome of CABOSUN is extremely exciting, as it marks the very first time that a therapy has shown a progression-free survival benefit over standard of care first-line treatment sunitinib for patients with previously untreated advanced renal cell carcinoma. Based on these findings, cabozantinib may have the potential to become a new gold standard for previously untreated patients following their diagnosis with advanced kidney cancer." In breast cancer, the combination of ribociclib and letrozole significantly improved progression-free survival compared with letrozole alone in patients with HR-positive, HER2-negative disease, according to findings from the Phase 3 MONALEESA-2 study. The improvement in PFS was identified at an interim analysis by an independent data monitoring committee, which recommended that the study be stopped. Following this analysis, Novartis, the developer of the drug, said it would now begin approval discussions with the FDA and other global regulatory agencies. The MONALEESA-2 data have not yet been released, but will be presented at an upcoming scientific meeting. Data will continue to be assessed for overall survival. A number of clinical trials are assessing ribociclib as a treatment for patients with breast cancer. The Phase 3 MONALEESA-3 trial is exploring ribociclib with fulvestrant for patients with advanced breast cancer following one prior line of endocrine therapy. The MONALEESA-7 study is currently exploring ribociclib with tamoxifen or goserelin, or a non-steroidal aromatase inhibitor, and goserelin for patients with HR-positive, HER2-negative advanced breast cancer. The European Commission has granted conditional marketing approval to daratumumab for the treatment of patients with relapsed refractory multiple myeloma previously treated with a proteasome inhibitor and immunomodulatory agent who progressed on their last therapy. The approval was based on findings from the Phase 2 Sirius MMY2002 study, the Phase 1-2 GEN501 study, and 3 other supportive studies. However, the approval is contingent on results from confirmatory trials. A combined analysis of the MMY2002 and GEN501 trials did show that at a mean follow-up of 14.8 months, the estimated median overall survival was 20 months with single-agent daratumumab in heavily pretreated patients. Daratumumab was granted an accelerated approval by the FDA in November 2015 as a monotherapy for patients with multiple myeloma following at least 3 prior therapies. The CD38-targeted monoclonal antibody has shown strong potential in 2 other clinical trials for multiple myeloma. In the Phase 2 CASTOR trial, which will be presented at the 2016 ASCO annual meeting, daratumumab combined with bortezomib and dexamethasone reduced the risk of disease progression by 61%. Secondly, the Phase 3 POLLUX trial also showed that daratumumab combined with lenalidomide and dexamethasone showed a 63% reduction in the risk of disease progression in patients with relapsed refractory disease. In gastric cancer, the combination of olaparib and paclitaxel failed to improve overall survival compared with paclitaxel and placebo. This was seen from findings in the Phase 3 GOLD trial for patients with advanced disease. Enrollment included 525 patients from China, Japan, South Korea, and Taiwan with gastric and gastroesophageal junction tumors who progressed after frontline therapy. The primary endpoint of the study was overall survival, while secondary endpoints were focused on progression-free survival, safety, and response. Additionally, tumor samples were tested for ATM expression, with 18% of the participants testing negative. Outcomes were found to be similar between the ATM negative group and the overall study population. Adverse events were also reported to be similar between the 2 arms. In a statement, Dr. Sean Bowen, Executive Vice President of Global Medicines Development and Chief Medical Officer at AstraZeneca, said, "The particular regimen in the GOLD study at a low dose and in combination with chemotherapy differs from other Phase 3 trials in the Lynparza program. We look forward to presenting the GOLD data and remain confident in Lynparza's clinical activity in a range of tumor types, including its approved use in BRCA-mutated ovarian cancer. Full results of this study will be presented at an upcoming medical meeting. That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.